So, in this session we are going to study about the one of the another major ecological function of phytochromes, that phytochromes are controlling the circadian rhythms. And I also in between I would like to remind you that try to follow with the textbook of plant physiology by Tays and Seiger, because you will be able to more closely observe with the figures and try to follow it, because it may not be possible to directly see from the projections, because the writings what you see over here may not be clear enough, and that is only what is possible at this moment from our side. But if you have any soft copy or a hard copy of this book of plant physiology by Tays and Seiger, you can equally follow it from the textbook more closely, especially with regarding to the figures that will help you to more understand in a better way. Here, this is the circadian rhythm with regarding to the diurnal movements of albizia leaves. The leaves are elevated in the morning and lowered in the evening. You must have seen the closing and folding of the leaves, and the leaves are elevated in the morning and lowered in the evening. So, it may be in an elevated position and it will be drooped position in the evening. In parallel with the raising and lowering of the leaves, the leaflets open and close. So, when it is in elevated position, the leaves will be open. When it is in drooped position in the evening, the leaves will be closed. So, this is happening based on the light and dark situation. Here you can see in the dark the leaves are open and the leaves will be in elevated position, and that is what you see over here. And in the dark time or in the evening, the leaves will be down in the drooped position. Also the leaflets will be closed, and this is simultaneously occurring. When light comes, again it will be opening, elevated, dark, drooped and closed, and this process continues. And now if you don't supply the light for a particular period, this rhythm continues but at a lower phase. See, the peak has reduced, and if you still continue in the dark phase, slowly the rhythm dies off, and ultimately the rhythm is totally stopped, and no more you can see the leaf is opening up. So, this is the what we refer to as, there are many of the functions taking place in plants based on this circadian rhythm, and since light and dark is involved, the light perception happens with the help of phytochromes. And what exactly happened? What is the physiological mechanism behind the opening and closing of the leaves? Here it is very clear depicted that we have at the base, at the petiolar region of the leaves, you have motor cells which are turgid. This is at open stage. And at the lower portion, dorsal cells, we have certain motor cells which are flaccid. So, this happens during the light situation when the leaves has to be opened. Leaflets are open, the ventral motor cells are turgid over here, and the dorsal cells are motor cells are flaccid. And the evening, the reversal of this happens, that is the ventral motor cells become flaccid and the dorsal motor cells become turgid, and based on that the leaflets close. So, this way this is basically happening because of the ion fluxes between the dorsal and ventral cells, and you know the osmoticum is building up, and the osmoticum is controlled because of the influx and efflux of the potassium ions. So, basically the potassium ion fluxes happens between the dorsal and ventral motor cells of this is being depicted in the case of Albizia pulvini, and this regulates the leaflet opening and closing, and the flux of these ions is being controlled by the phytochrome. And here you can see how exactly the phytochrome is going to influence these particular actions. Here you can see from the figure that phytochrome beyond the reception of light, red light gets converted into phytochrome PFR form, and on reception of R red light it goes back into the PR form. So, phytochrome B is synthesized in the cytoplasm in the inactive PRB form. On reception of red light, it gets converted into PFRB form. When converted into the active PFRB form by red light, it moves into the nucleus. Only this active form can move into the nucleus because this is the active form. And once it reaches the nucleus, the PFR binds to a dimer. See here, the dimer of the transcription factor, that is PIF3, PIF3, which is bound to the G box. So already this PIF3 portion, this is a dimer form attached along with the G box. So here the PFR entering inside goes and bind to that. Upon addition of the pre-initiation complex, on binding of that you can see the pre-initiation complex, that is the PIC comes and binds to the TATA region, TA region, and based on that the transcription process starts. The transcription of MY genes initiates and MYB protein is produced. And MYB protein is again a transcription factor. This will come and bind. MYB transcription factor in turn activate the transcription of other genes, especially one of the genes described is LHCB. LHCB is nothing but light harvesting complex protein. So on the binding of the MYB protein, the light harvesting complex synthesis, the protein synthesis initiates. Again I can explain once again. The phytochrome B synthesized is in inactive form. On reception of the red light, it gets converted into PFR form. This is active. This moves into the nucleus. This binds to the dimer form of PIF3. PIF3 is nothing but this is the that is the PIF region. This is bound to the G box, and on binding of PFRB to the PIF3, the PIC, that is the pre-initiation complex, comes and binds to the TATA region, TA region, and this initiates the synthesis of MYB, the transcription factor. The transcription factor now comes and binds to the gene responsible for the synthesis of LHCB, that is light harvesting complex, and that initiates the synthesis of LHCB. So this is how the phytochrome is going to control the gene expression of light harvesting complex. This one example is shown. Similarly, the phytochrome can control many of the gene expression. Also, you can see in the next slide the detailed view of how the phytochromes can control. Although this looks to be more complicated, a detailed analysis will tell you how the phytochromes are going to control the light regulated gene expression. I will explain to you some examples from here, and it will be more better to follow from the text directly. Here on red light reception, the phytochrome PR form gets converted into PFR form. PFR form on along with the expenditure of energy, that is ATP, that gets phosphorylated. PFR form is phosphorylated. This can directly move into the nucleus and regulate some of the gene expressions. That is one way of gene activation can happen. Another way of gene activation is that PFR form can phosphorylate PSK. PSK is a phytochrome kinase substrate. On phosphorylation of that again can regulate some of the gene functions. Now another route is that the phytochrome on phosphorylation, PFR form on phosphorylation, can act upon the G protein. G proteins can further through secondary signaling intermediates like CGMP, that is guanosine monophosphate, or another secondary signaling agents like calcium carbonyl complex can move inside the nucleus and finally regulate some of the gene expressions. Another possibility is that the PR form, this is with reference to PRA form, with reference to PRB form, that is phytochrome B, that is type 2, that gets on reception of red light, that gets converted into PFR. PFR can further gets phosphorylated, and PFR after phosphorylation again it can influence the G protein and it can continue in this path of secondary signaling through cGMP or calcium cadmium complex, enter the nucleus and regulate the gene expression. Or otherwise, the PFRB after phosphorylation can directly move in, and through the PIF factor, which we already seen in the previous slide, it can regulate the gene expression. Or in other way, it can regulate through nucleoside diphosphate kinase NDPK2. It can activate some of the gene functions. It can regulate some of the gene functions. And towards this side, we have that dark regulation happening. The dark regulation occurs when there is, this all happens when there is light. But when there is dark, the COP protein is synthesized, it moves inside, and it can degrade the HY5 protein, and that can repress some of the gene functions. And when there is light, the degradation of COP protein take place, and it may not be active anymore, and the influence of phytochrome will occur. So this is a more complex area where a detailed elaboration is given on how the phytochrome, either in the A form or in the B form, can regulate many of the gene functions, either directly or indirectly through G protein and secondary signaling through cGMP or calcium cadmium complex. So I suggest that you follow with the textbook and understand in a more better way how phytochromes can directly influence the gene expression levels and influence many of the physiological functions, and understand in a better way. So we have come to the end of this session on phytochromes, and hope you have understood it. And if you have any clarifications, my email is available. You can either write in my email ID, and I will try to give you more clarifications on this. My email ID is jtputhur@yahoo.com, jtputhur@yahoo.com, and I will be more happy to give you more clarifications. If you have any clarifications, you can write to my email ID. Thank you.